The other big data that is very commonly generated as well as used is transcriptomics, and as the name suggests and as we have discussed before, transcriptomics is the overall component of RNA transcripts that are produced by the cell. So what is the main reason for taking up transcriptomics? As I mentioned, genome is something that is common across all the cells, let's say, of a human body. However, transcriptome is variable, as well as differentially regulated to bring about specific functions of a specific cell. So as we were discussing before, a skin cell or a liver cell or a neuron or an epithelial cell would have the same genomic content, would have the identical DNA in a human body. However, their functional differences, the differences in the function that they do, like skin cell being a skin cell, liver cell being a liver cell, those functional differences are brought about by how the DNA is transcribed, how many of the genes that are there in the DNA, let's say 30,000, how many of those are transcribed and made into functional proteins in different cells. So a set of genes that will be on and transcribed and producing proteins in skin cells will be, that list will be different from the list of genes that are on producing the transcript and producing the protein. So the main purpose of doing transcriptomics is to understand this complex relationship which comes from the DNA and which results in the phenotypic change. And as I mentioned, each of the cell type can have a different phenotypics. And this is done at a genome-wide level for all the genes that are present in a cell. So what are the various processes or various results or various questions people ask when they are doing transcriptomics? First, as we just discussed, when and where are the genes expressed? So for example, in a liver cell and a skin cell, as we said, there will be differences in the list of genes expressed meaning producing RNA and then producing protein, and from the list here. Okay, that is one thing, that is the difference between the two cell types. Now if I want to see, for example, what happens in a liver cell when I am fasting versus what happens when I am fed, that list also will be different. So when and where, when in the same tissue, when are certain genes switched on, when are certain genes switched off, depending on what condition the cell is in, or in this case body is, where they are switched on, like whether it's in liver or in skin, right? There are certain genes that are only switched on in certain specific organs or cells. They're not switched on anywhere else, right? So for example, liver cells would produce liver enzymes. That is the function of liver cells, one of the functions of liver cells. Skin cells, even though they have the genes that produce the same liver enzymes, they never produce the liver enzymes, right? So that's what is when and where. The other thing was how gene expression level differ across cell types and states. So cell types I just mentioned, types are skin and liver, states could be fed and fasting states. So sort of similar question. So it could be that in fasting and fed state of a liver cell, there are certain genes that are changing, there are certain genes that do not change. So what are those genes that change, what are those genes that do not change? That's another question that we would like to address. Now, The question that we want to ask also is, what is the function role of different genes and what cellular processes that they are involved in or participate in. So if you think about it in sort of genome wide scale, right? So what does this uh, genome wide scale, what does the transcriptome give us? It gives us the all the genes that are expressed. So out of 30,000 odd genes, what are all the genes that are expressed in this particular state or a type, and based on that list of genes we can start doing analysis to figure out what are the cellular processes that are switched on in this particular cell type or at this particular state, right? So it allows us to make from the set of genes that are expressed what cellular processes are on. Whether, let's say for example, uh, glucose metabolism is on, or lipid metabolism is on, or fat metabolism is on. So those kind of cellular processes can be also figured out. You can also figure out other specific processes, say for example processes involved in cell cycle or processes involved in nucleotide synthesis. So all those things can be figured out by studying the list of genes that are expressed in different cell types, and therefore for the genes that are expressed and are involved in a certain process, and there was no annotation for that particular gene, we can actually start assigning roles to those unknown function genes. Transcriptomics also gives us an idea of how these genes are regulated, because as I mentioned before, let's say this is your gene, this is the start and stop of a gene, start, stop of a gene. Here is the region which is the promoter region, and promoter region is the where on off switch is. So by looking at what all genes are switched on under certain conditions, you start figuring out how these set of genes that are switched on are regulated, right? So we can do experiments to figure out the proteins that come and bind here to switch this on. If we remove that particular protein, all the genes that are regulated by that particular protein will be not switched on. So we will know the function of how that particular set of genes are switched on. We also get to know when two or more genes are interacting, how do they interact. So for example, if I have gene 1, when it is present, I know what is the list of genes. When gene 2 is present, I know the list of genes. Now I can ask the question, when gene 1 plus 2 is present, what is the list of genes? So that allows us to see how these genes interact. And when you have these kind of information and you compare between normal and a disease, or a normal and a disease and a treatment, you start seeing what are the gene expression lists that are now aberrant in a disease state versus a normal state. And when you give a treatment, whether that aberrant list becomes more like normal, right? So this is how you can actually see what are the molecular consequences or what are the functional differences that come about when diseases happen. So say for example, normal cell versus cancer cell, right? If I do transcriptomic of both, I'll see a lot of genes that are either upregulated or switched on in cancer, which were not switched on in normal, or switched off in cancer, which were not in the normal. Now I know what, and therefore I will be able to figure out how is this cancer being manifested, what is the molecular cause of this cancer. So if I now target drugs specifically to these particular pathways, which will be the treatment, I can then go and ask whether the treatment worked or not. And this is one of the ways many of the cancer drugs are being identified and sort of taken to clinical trials. And so you see, genomics doesn't give all this information, whereas one readout, the readout from genome, which is the transcriptome, allows you to ask so many different questions, and by combining these questions, all of these questions or some of these questions, we can start figuring out the function of the gene, the function of the genome, the function of the state of the cell, function of a cell, and all those things. So these high throughput large scale assays allow you to measure and track many genes or their protein products, but where transcriptomics is mainly genes, in parallel in different conditions. So you can actually compare normal, as I said, versus cancer versus treatment, right? Treated. So this could be hundreds of samples here, hundreds of samples here, hundreds of samples here. This will give you a huge data set with enough power to ask the question, does the treatment work in treating this cancer? And this is normally how we approach a particular problem. And this could be any disease actually, right? So what are the major ways of doing transcriptomics? We'll cover that in the next few slides. So analysis is something like this. So this is just to give you a flavor of how we know that certain genes are switched on and off. So in this case, red color indicates that it is switched off or what is called down regulated. So this is red, up regulated is green, right? These are the various bacteria, oh sorry, other way around. Down regulated is green, up regulated is red, right? These are the different bacteria, these are some of the bacterial strains. These are the genes that are in humans when an infection happens. So you can see that, for example, these bacterial species switch on a particular set of genes here, which is not done by another set of bacteria. Like these must be lactobacteria, these are bacillus bacteria. So these bacteria switch on a set of genes which are not switched on by sort of a benign bacteria. So if this is a pathogenic bacteria, we know exactly what the body, how the body is responding to this pathogenic bacteria, right? So this way you can actually figure out that what are the functional pathways that get affected when a particular bacterial strain infects a human body. And that is exactly what we normally try to get as a result. And this kind of visualization is called heat map. And this is not a heat map, this is a visual of a microarray, which we'll see in a bit what microarray means. Each spot is a particular gene, so you have either bright fluorescence or low fluorescence, and you can actually, and whenever there is black, that means there is no fluorescence here. So this is sort of dense, equally spaced spots. Whenever there is dark, there is no expression of a particular gene. So as I was mentioning, transcriptomics is the first step of how the genome is read. That has a lot of information, and that information gets translated to function more clearly or to the phenotype more clearly than what genome will tell us, because genome is identical in all the cells of a human body. But because of this sort of architecture, right? What are the switch on and off circuits, right? What is the network of control of various genes? What are the regulators or regulatory elements that are involved in switching on and off or slightly changing the expression level of that particular gene? On top of that, we have overlapping genes and mobile genes. Even if we ignore that, these two processes make the prediction from DNA to RNA to function or phenotype harder. Okay. So, when we were talking about RNA molecule, primarily we were talking about messenger RNA, which is the RNA which is this one, which is produced by the DNA. There are various other types of RNA which we haven't covered. This is involved in translation. This is part of the ribosome, which we discussed earlier, and these are other non-coding RNA. These are non-coding RNA, which we won't cover right now. Primarily, transcriptomics covers this mRNA. So, what are the main methods of generating transcriptomic data? And as I was when we were discussing about genomics, there are two technologies, one which is an older technology, one which is a more recent technology. So, in DNA sequencing, we had Sanger method and we had NGS methods, and in NGS we had Illumina method, right? In transcriptomic, genome-wide transcriptomic data generation, we also have two methods. One is an older method which is called the microarrays. Another one is a next generation sequencing method which is called RNA-seq. So, the major difference between the two methods is microarrays is essentially a silicon wafer on which there are sort of attached to the silicon wafer small DNA fragments of a particular genome. So, if I were to talk about human microarray, small fragments of human DNA are attached in a grid on this silicon wafer, and therefore, when you take a human sample and put it on to this after labeling and fluorescence and like after some experimental steps, you will see some spots lighting up, some spots not lighting up. The spots that light up means those genes are expressed, the spots that don't light up means those genes are not expressed. So, this requires prior info on the DNA sequence of an organism that you want to make microarrays of. And whereas RNA-seq, which is based on NGS technologies, doesn't have that requirement, and it is more digital because you can now count number of transcripts that are produced. We'll see that in a bit. So, microarrays, essentially the process between how a microarray experimental steps are done and how a next generation sequencing experimental steps are done is practically similar. So, you take two samples, one which is a normal sample and let us say the other one is a tumor sample. You fragment it and you label the fragments with a fluorescent marker called biotin, and this is a microarray. It's made by a company called Affymetrix. And when you load these biotin labeled samples on this silicon wafer which is in this window, right, and you then process it through its specific machine, what you will get are these kind of images. So, once you scan it, you will get on the screen, you will get this kind of an image. This is an image of a tumor, this is an image from the normal. Now, this is a very high density array of, so if you remember in the previous one, this was the sort of, this is, you can think this is sort of is a zoomed out version of the microarray. Usually if you look at a microarray screen scan, this would how it would look like. Now, there are specific dots which are zoomed out here which give level of expression. So, if it is high expression, then it will have high intensity. If it is low expression, then it will have low intensity. So essentially you will have something for the tumor, something for the normal. You can compare. What you will see that there are certain genes that are commonly expressed, which is expected, but then there are certain genes which are not expressed in one case and expressed in the other case, and those are the differences that you want to now figure out. And there are computational methods of doing it. You don't do manually compare that, because so the other thing that you get here is you normally get tumor over normal ratios. So if the ratio for a particular gene is greater than 1, that means it's over expressed in tumor than in normal. If it is less than 1, then it's a reverse. So you always get relative abundance, you do not get actual abundance, right? And that's one of the advantages, that's one of the sort of lacune in the microarray technology. However, microarrays are very cheap to use. If you are working with an organism which has a microarray available, that would be the cheaper way to go about it, and it's faster, less processing compared to next generation sequencing method, RNA-seq. So here is the process again. So you have your normal sample, you do RNA extraction, you make cDNA out of it, then you make by using reverse transcriptase. So in central dogma we had DNA, which was doing RNA polymerase to RNA, but we now have another enzyme which calls reverse transcriptase, which will convert RNA to DNA. So when you start with RNA, you create DNA, and this is called complementary DNA or cDNA. Once that is done, you amplify that and you load it onto the microarray, which has these, which has a grid. In this case, this is being shown as a grass. Then you can actually find these kind of images, compare these images between the two samples, and you'll get and analyze those intensity samples to figure out what are the differences. So microarray would actually allow you to not only detect the whole gene, but also detect different splice variants, right? And this is something that we can actually use to see whether there is a splice variation difference in between the two samples as well. RNA-seq, on the other hand, is based on next generation sequencing technology like Illumina. We have discussed how Illumina works, so we won't go into that detail, but again the steps are essentially the same. You take RNA, you isolate RNA, that means remove DNA contamination. You can either do poly A selection, which means it would only take separate all the genes that have poly A tail, or you can do what is called ribodepletion, which would mean all the genes that are being actively translated by the ribosome are removed, or you can do just a general size collection. So any of the three methods can be used. Then reverse transcription is done to create cDNA, which can be then used like normal DNA sample as we discussed before, which means they need to be fragmented, then tagged, then adapters are attachment, and then sequencing. And similar to microarrays, you get information regarding all the splices, whether they are present or not, right? And so you can detect whether they, in this case, exon 1, 2 and 3 are together. Here it is exon 1 and 3 only. So you can get all these splice variants in the genome in one go. But this part is different. After you have converted into DNA, it just essentially is the next generation sequencing that we did in the last in previously when we were discussing DNA sequencing. Now, next generation sequencing is based on 2-3 different technologies, and there is a reason for that. So, when we were discussing Illumina protocol, we said the maximum range is around 150 bases that it can generate in one go. While that is a slight limitation, but it compensates that short computationally intensive process by generating a large amount of data and allowing us to build alignment maps, which we saw previously. So, whereas Pacific Biosciences can have up to 500 bases to 5 kb. So, these are called long read, this is called short read, and this is also long read. Again, here in this case, long read, the way rather than being linear, these molecules are circular, which means that now it can, that plasmid kind of a shape can be longer than 150 bases, and therefore you can sequence the whole sequence and whole circle to get you the long size DNA fragments. Both of the technologies, Illumina and Pacific Biosciences, based on fluorescence. However, Oxford Nanopore is another technology which doesn't rely on fluorescence molecule, but on the charge on each of the nucleotide. And the way these technologies are developed, that the DNA is passing through a narrow channel, and as it is passing, as different nucleotides keep on coming in, they change the charge on this molecule, and that change in charge is detected and results in a generation of a graph, which we know has a very high confidence in calling each letter very precisely.